Oh, I can turn it off. That's what you mean?
I don't know, just when you loop it.
Okay.
I'm just waiting for witness.
Yeah. Okay.
It's making me use Google Chrome.
Facebook or YouTube?
And he came to Maryland to visit Daphne Suarez, who he was working with as she did research down there. And she had brought him up here, and he came by to talk about fish. And I could tell immediately that he was a very passionate person about all the things that he did. And he was wanting to do a lot of genetic work in fish, and there weren't really any fish genetics people in Ecuador. So he found an insect lab to work in, showing his first signs of determination. And he gathered samples from some of the national parks. And from time to time, he would send me an email about how he was doing. And so this is, and he looks so young. And he would explain to me, what he was doing and how it was all going. He was collecting fish both in the daytime and at nighttime, taking lots of pictures, and already doing dissections. So that was young Danny. And he would also, he turned that paper into those data into a paper and a poster, so he sent me a copy of that too. So he made good progress during that undergraduate thesis. So then I was fortunate in 2014 when he decided to come to University of Maryland for graduate school. And he was only able to come because he was determined enough to get a doctoral fellowship from the Secretariat of Education, Science, Technology, and Innovation of the Ecuadorian government. So that's who has supported him while he's been here. So when Danny arrived, I asked him what he wanted to do, and he said, well, I want to do behavior. And I said, well, that's great, but I don't know anything about behavior. He said, well, that's really what I want to do. And so I said, okay. And so much of the behavior work you'll see today is stuff that he really did from scratch by himself, and I would kind of watch from time to time and say, well, that's great, really interesting. Let's turn that into something, a cool paper. But I also convinced him that since he was in a genetics lab, he really should do some genetics. And so you'll see today his thesis is kind of a marriage between those two different approaches. So I've always been impressed with Danny's artistic skills. He has a really great flair for that. And so one of the first things he did was design us a lab logo when he came. And the first year we wrote a review. And so these are some of the beautiful figures that he made for that review on cichlid sensory systems. So I think his artistic perspective really comes through and makes him a stronger scientist. So partway through his degree, he wanted to go back to the tropics, and so we decided to participate in a field course on animal communication that Jerry Wilkinson and Cindy Moss were doing with a group from University of Tübingen. And there, Danny worked with Victoria Ferenc. So here we are in the lab, and we're also out fishing for the predator Cichla monoculus. And we were able to catch it, but only because we threw ourselves on the mercy of everybody on the island to try and help us. This was the hardest fish to catch. So we did succeed. This is like a 2-foot long fish. I mean, it's really big.
Wow.
So he was successful in doing that. So during his PhD, Danny has been very successful. Before he came, he actually had 2 papers, one in current biology. And since then, he wrote 1 review, he helped with another review, and he has 3 data papers published. He has another paper that's in revision right now, and he has a couple more nearing completion. So he should hit that 10 publication mark pretty soon. He's also been participated in the University of London Sensory Ecology course. He was awarded the Smithsonian Research Fellowship, so he went back to Panama for another 3 months of work on his favorite characins. And he's given presentations at meetings all around the world. So I'm really happy that Danny was so persistent and was able to come to the lab. I really love having students who come and have a really different perspective and broaden sort of the way I think about things. And so it's been really great to have him here. So here he is on a recent trip with Miranda to visit Pratima. And with that, I will turn things over to Danny, who's going to tell you about proximate and ultimate insights into the evolution of color vision in tropical freshwater fish.
Hi. Hi, I'm Danny, and I'm going to talk about my dissertation research, which is about that. Thank you, Karen, for the introduction. So let's think about a little bit about color in nature. So coloration, a lot of animals, you have animal coloration, and they use this for several behaviors. And in order to detect also animal color patterns, animals need to have color vision. So color vision in behaviors mediated by animal color patterns are also, like, for example, prey and predator detection, also inter and conspecific communication, as well as foraging. And also animal coloration patterns are also involving sexual selection, as animals find mates that way. So just a quick reminder of how vision starts. So vision starts when light hits the eye, it goes through the cornea, and it goes through the lens, and then it hits the retina. And in the retina we have this range of that has been called the retinal mosaic. And here is with this chemical signal gets transformed into a neural signal, and then it gets sent to the brain for the animal to make a decision. In many fish, photoreceptors are arranged in a retinal mosaic where double cones, this is what we call them, 4 of them are surrounding one single cone. And also, you know that cones are used for diurnal vision, meaning kind of bright light conditions, and rods are used in night vision, meaning dim light conditions. So photoreceptors have visual pigments, and these are the ones who are sensitive to light. So a visual pigment is made of an opsin protein, which has 7 alpha helixes, and it has an extracellular, transmembrane, and cytoplasmic region. And then the light-sensitive molecule, the chromophore, or 11-cis-retinal. So when light hits the chromophore, it photoisomerizes to all-trans retinal, and this also causes a conformation change in the opsin protein, and then the phototransduction cascade initiates, which ends in a visual signal. So usually an opsin absorbs light on a particular part of the wavelength spectrum. So here you have on the x-axis the wavelength, and on the y-axis the absorption. And you can see that this pigment, for example, absorbs light at 450 nanometers, kind of. So scientists use for measuring maximal absorbance of visual pigments different techniques. However, one technique that is used a lot is microspectrophotometry, or MSP. And it's basically, you take the eye of an animal, and you dissect the retina, and you isolate the photoreceptors, and you put them here in your slide sample. And then you shoot kind of like a laser beam from this xenon lamp, and then it gets refracted through here, and it goes through here where your sample is, and then light gets collected in the photomultiplier, and then the information is sent to the computer. And usually what you do is that you shoot the laser. Once you find a photoreceptor in your sample, you shoot the laser next to the photoreceptor, and then right through the photoreceptor, and then you compare those two light absorptions, and then you know, like, oh, this is the spectral sensitivity. So opsin proteins are coded by opsin genes, and we have different opsin classes, which are the result of several duplications early in the history of vertebrates. These occur around 450 million years ago. So this single opsin gene duplicated these different ones, giving origin to these different opsin classes. Notice how, for example, there are duplications. For example, humans have 2 red opsins, and they don't have a blue opsin or a green opsin, for example. However, they do have a UV opsin, or SWS1. And for example, lamprey has 2 RH2s as well. So these different opsin classes are really important because they have unique amino acid sequences. And each of these amino acid sequences interact with the 11-cis-retinal, or the same chromophore. If you see from above, you can see how uniquely they interact with the light-sensitive molecule, the chromophore. And what happens is that these different interactions of the amino acids leads to different shift the maximal absorption of the protein. So here you have how these different opsin genes create different visual pigments that absorb light in different parts of the wavelength spectrum. For example, UV, blue, green, or red. So now that we know all this, so what are the requirements for color vision? You need to possess more than one photoreceptor type, meaning that you must have two photoreceptors that are spectrally different. And then you must have a mechanism in a way that you can compare these two signals, at least. And now fish are really good for studying color vision. They have a very rich phylogenetic history, a lot of different species, a lot of different morphologies, and they live in a variety of places as well. And they have a lot of adaptations to live in the underwater environment. And the underwater environment also puts another layer of complexity into visual systems because of the physical-chemical properties of water. Light gets transmitted in a different way. So based on the composition of water, the spectral composition and the light intensity of the water will vary between different ecosystems, and this will have an impact in the visual systems that animals live, where the animals have in order to live in this environment. So, So here I guess all of you know about cichlids. They have a kind of a Gondwana distribution. They are very diverse. They are a prime example of adaptive radiation and fast speciation rates. And they are very pretty. A lot of labs studying them, and previous research in the lab has focused on Lake Malawi. So Lake Malawi is in East Africa and is a very clear water lake. You have here more than 500 species of cichlids that have radiated really fast. And previous research in the lab has characterized the genetic basis of the visual system of these fish. So we know that they have 7 different opsins in their genomes, and each of these opsin genes codes for visual pigments that, as you can see here, absorbing a different part of the wavelength spectrum. So they have basically visual pigments that cover the UV, the violet, the blue-green, different greens, and the red part of the wavelength spectrum. However, something that is very interesting about African cichlids is that they express differently these sets of opsins that they have. So for example, we have this first group, and they express the UV opsin, the blue, green, and the green. And then you have other groups of cichlids within the same lake that are similar to the first one, but they are different in that they express the violet opsin. And then you have this third group that express the blue or the red opsin. So what happens, you have here cone opsin expression, sorry. And then what happens is that this different opsin expression ends up in different visual pigments that these fish have. So you can see here how this group of fish is sensitive to the short part of the wavelength spectrum, this one is sensitive to the middle part, and this one is sensitive to the long part. So this all creates a lot of variability about the visual sensitivities within the lake. And we have termed this as visual palettes, so we denominated this as the short, medium, or long visual palettes. So for my thesis, I wanted to try to understand how color vision is evolving, and different ways in we can see what is happening and how it has been, what is the adaptive significance of this. So for this, you're going to see data on cichlid behavioral color vision, on the visual system of Amazonian cichlids, and visual systems of neotropical galaxiforms. So, and I wanted also to try to understand, like in a bigger picture way, like what are the proximate mechanisms for color vision, in terms of like what is the interaction of photoreceptors between cichlids, and which opsin genes are being expressed in other fish as well, and what spectral tuning mechanisms fish use, what do they use in order to like adjust their spectral sensitivity to different parts, and also what affect the diversity of visual pigments. And then from an ultimate point of view, what is the adaptive significance of color vision? Like, do cichlids have color discrimination, or do fish visual systems adapt to the light environment? And what are the evolutionary dynamics of opsin genes and these like throughout the phylogenesis? So now that we know a little bit about Malawi, I wanted to understand, so we have now these opsin genes, we know that these fish express these different opsins, and they have these visual pigments, and I wanted to understand what does it mean behaviorally, what does it mean for the fish to be able to have these different sensitivities. And we focus on this fish that is Metraclima venetus. It's a rock-dwelling cichlid from Lake Malawi. And here is the visual system. Here is opsin expression, and now we know which genes it's expressing. And this, as you know, produces these different visual pigments, and that we actually know the lambda max, their maximal absorption, so that we know that they absorb at that part of the wavelength spectrum. And then I wanted to train them, I wanted to make the fish discriminate between different colors. And for this, we created different stimuli. We printed in paper, and we literally just measure with a spectrometer the reflectance of these colors. Here you have on the y-axis reflectance, and you can see the different reflectance profiles of each of these different shades of blue, yellow, and gray. And I wanted to train the fish in order to discriminate blue from these different colors. You can use these data that I just showed you, the visual sensitivity is one, the light environment where they live in, and also these reflectance profiles in order to calculate quantum catches. They represent the number of incident photons that each visual pigment are receiving. And so with this, you can calculate, you can do visual models. And visual models help you understand how animals are comparing signals. So here you have the three different, we have also termed them as short, middle, and long, because based on where in the wavelength spectrum they are. And here you can see, for example, a model assuming two signals. So this assuming quantum catches in the x-axis only from single cones or short for the short pigment. And here you have on the y-axis the middle and long quantum catches. You can see that there are different colors that we saw before. Here they plot gray is overlapping with blue. So for example, according to these visual models, fish maybe would not be able to discriminate these two colors. But also, we can assume that fish, that cichlids are trichromatic, meaning that they are kind of like humans. And you can see in this plot, each axis represents a different photoreceptor. And here you can see how our colors actually spread a lot. And according to this visual model, this visual color space model, you can see how colors should be discriminable. And this is taking into account that the retina is receiving a 3-signal input. So we're going to put the visual modeling aside. Here you can see we housed different fish in individual tanks, and we started to train them for blue. So we attach, we put food here, and we attach this tube, and then we first train the fish to tap the feeder tube in order to release food. And then we attach this blue color, this blue circle to it, and we train the fish to tap that blue color in order to receive food. And then through second conditioning, we took out the feeder tube and we just put a color card. And then we trained the fish to tap the color card. And then finally, they were ready for testing. This is how testing was like. So we did different behavioral experiments. One was a 12 alternative choice test. Here we try all the possible combinations of the different shades of blue and different shades of gray and yellow. And then we did a multiple choice test where we showed different also blues, but with different shades of gray. So we did for these different combinations of color cards for blue, dark blue, and light blue. And then we finally did a third experiment, which was a multiple choice test, or a color bias test, because I wanted to see if, besides discriminating from blue, these fish were biased towards some color, as some fish have bias for some colors. And this is kind of like what it looked like. So you can see that the fish comes, and then it taps the stimuli. It's pretty straightforward. They can be really fast. And this is experiment number 3. And after that, they will be rewarded. So these are the behavioral results. So each box is a pairwise combination, and on the x-axis you have each of them is a different fish, and on the y-axis you have the proportion of correct choices. So notice that overall, fish significantly chose blue over the distractor stimuli in all cases. Here they have a little trouble with light blue and grays, but overall all of them are pretty good at discriminating colors, which actually is in agreement with our visual modeling hypothesis that their visual system is comparing 3 spectral signals. Now for the results of experiments number 2, here you have all the results for blue, light blue, and dark blue, and you can see that also fish significantly chose blue over the distractor stimuli, pretty good. And we see something similar here on the multiple color test. You can see, however, what was interesting, something that happened is that fish made a few mistakes, as you can see here. We prevented them from getting like a 100% score, And 80% of these mistakes was with purple. And I guess it makes sense because purple has a little bit of blue, so basically fish were confusing blue and purple. And this made us think about the color limitations of cichlids, and I wanted to understand more about what are the limits of their color vision and how we can try to understand that. And for that, you do color thresholds experiments. So color thresholds is what limits our capacity to discriminate in different colors. So for some of you, you can maybe you all already know that here there are different colors, or here there are 5 or 6. Actually, we have like 3 in the second and third row, which suggests that also our visual system vary in our capacity to discriminate colors. Color limitation also arises when you have different matching backgrounds. So for example, here maybe for me, like it seems that these ones at the sides are all very similar. However, they are all different colors, all the colors here. So this is just to give you an insight that color thresholds determine our capacity to discriminate colors. And for studying color thresholds, you have to understand that they are defined by receptor noise. And this is neural interference that arises in the retina, as well as in different, maybe in higher processing parts of the brain of the animal. Photoreceptors noise determines the smallest chromatic distance between two colors, between one pair of colors. And the units of this chromatic distance are GNDs, which are called just noticeable differences. Just to give you more like how does it work. For example, here we have blue comparing to all of these different colors. And here you have on the y-axis the chromatic distance. And you can see that when we use a noise value of 0.01, we have large chromatic distances of blue versus yellow, blue versus brown, etc. However, if you increase a little bit the noise, chromatic distance decreases and colors start to be less discriminable. So when it is more noisy, chromatic distance decreases. So with this, we started working in the lab with Michaela Taylor. And then here you have the reflectance profile. So we printed a lot of more stimuli. And here you have the wavelength spectrum. So here we have our blue that we did originally, and then we added red and more red, and we designed different stimuli that differ in the reflectance profile. So as you can see, the distractor stimuli number 7 is the most different from the blue. And then you can put this in your visual color model, and you can see again that here is blue, and distractor color number 7, for example, is here. So even already in the color space, we can see that these are distant colors. And then we did 2 alternative choice tests, basically our rewarded blue and all the different colors. And then while we were testing, we realized that there was a threshold here, meaning that fish started to choose blue over the distractor stimuli more after these colors. So we designed a brightness test for this part. So here you have, this consisted in color cards where there was the blue and there was the distractor stimuli number 3, for example. And we found different colors that are chromatically similar to distractor color number 3, but also that they differ in brightness. So you can see that there are darker shades and lighter shades as well of these colors. And we did this for distractor stimuli number 4 and distractor stimuli number 5, where we thought where the threshold was. And here you can see how it went. So fish did good, but they had a lot of difficulty. So here we have the behavioral results. So remember that chromatic distance units are GNDs, and also usually values below 1 GND, according to these models, suggest that values that these colors are not discriminable. So here you have on the x-axis the chromatic distance. Here we have our rewarded color blue, and here we have the different distractor stimuli. Notice how, for example, distractor number 7 is the most chromatically distant again. And here on the y-axis you have the proportion of correct choices. Each black dot is the proportion of correct choices of blue over the distractor stimuli. And notice how they start to reach significance after stimuli number 4. And now this is confirmed by our second experiment. Again, you have chromatic distance here, and notice how right after stimuli number 3, fish significantly start to choose blue over the distractor stimuli again. So this second experiment was interesting because it backed up the findings of our first experiment. However, notice that the threshold is around 3 GNDs. The behavioral threshold is around 3 GNDs, and it's not around here. So what this suggests is that noise is higher than we thought it was. So this noise, this new value, which was 0.16, the new value is 0.16, and this allows us to calculate more, to think about more the visual ecology of Lake Malawi. So for this, we wanted to compare how color contrast happens in Lake Malawi. So here we have the blue color of Microclima venetos, and we compare against the rock, green grass, and orange algae, and also space light. The space light is basically just horizontal light versus the water column. So how blue will be compared against those backgrounds, and then also how blue will be compared against different colors from different fish species in Lake Malawi, like yellow, black, green, and orange. So here are the results. So on the x-axis you have chromatic distances, and you have for backgrounds first and for cyclic colors. And you can see that the highest chromatic distance is between blue and space light, Otter and Maulamba Bay. Those are some different beaches in Lake Malawi, and you can see how it's very contrasting that instead of like red grass or orange rock, for example. And then also how there are high chromatic distances of cyclic colors of blue versus yellows, for example, than blue versus black or so. And these large chromatic distances between colors are in agreement with the behavioral ecology of Lake Malawi in cichlids. So in the wild, female and venetos swim above the rocks of male territories, and males swim up to them and to perform courtship displays, and if he gets lucky, the female follows him back to his territory to mate. So I guess it makes sense for the females to be able to discriminate the blue male from the water column, as well as the blue male from different other males from different colors. And this is also interesting because blue and yellow nuptial colorations have been acquired repeatedly in Lake Malawi. Here we have a study where they analyze the phylogenetic relationships of different species, and you can see how sister species here, they start to develop nuptial coloration differences between blue and yellow always. So the first column are males, and the second column are females, and you can see how always sister species start to develop these differences in nuptial coloration. So what we have learned is that cichlids can be trained to perform color discrimination tasks. The cichlids may successfully discriminate rewarding stimuli from a variety of distractor stimuli. We successfully determine their color thresholds and receptor noise, and the visual modeling suggests chromatic distance is higher between blue and yellow and between blue and space light, which might be important for mating. So now that we know more about what are the behavioral implications of these different spectral sensitivities, I wanted to understand more about what does it mean for other major cichlid lineage, which is the neotropical cichlid lineage, and we wanted to understand how it has changed in comparison to the African cichlids. And for this, we worked with Cesar Martins and Erika Ramos in Sao Paulo State University, and we analyzed the genomes and transcriptomes of 4 Amazonian species. So here you have, what we found is that Amazonian cichlids have a less diverse complement of visual pigments than African cichlids, which is something different than we expected. So here you have the 4 species that we analyzed, and here you have each different option. Here you have tilapia, which is an African cichlid, and you can see how Amazonian cichlids have lost the RH2B opsin, and also how they have lost other opsin classes. So this is interesting because even though they are phylogenetically distant, they all have converging losing different opsins. And then if we analyze amino acid sequences of all of these opsins between these 4 species, we also found that the SWS2A opsin, here is the number of amino acid substitutions in the y-axis, here we have the opsins. This is the most variable opsin in amino acid variation. In terms of like, here we have in solid parts the transmembrane region, exclusively this one, and then also in chromophore binding pocket sites, which is amino acids that may have a big impact in shifting sensitivity because they are facing the light sensitive molecule. And this is interesting because this differs from previous findings from other fishes, because they usually found most of this variation in the UV opsin. However, we are finding this in the blue opsin. This suggests also that this gene may be under positive selection. And then if we analyze their cone opsin expression, here you have on the y-axis of these 4 different species, you can see that they all express a similar opsin set, let's say, blue, green, and red. And it's very interesting because they are in the phylogenetic tree, they are in different tribes in the cichlid lineages. However, they all converge on that. And now, like if you remember the different visual opsin palettes that African cichlids have, I guess you can develop your intuition about which one is similar to the Lake Malawi palette, which is the long palette, right? Which is also present in African cichlids. And this is cool because now we can think about adult opsin expression in a phylogenetic context. So here I have coded blue for the short palette, green for the medium palette, red for the long palette. And here you have in this part of the phylogeny cichlids from the African lakes, and here you have cichlids from neotropical rivers that we know adult opsin expression of. And you can see that neotropical cichlids, they all express long palette. However, African species are the ones who exhibit most of this variability in opsin expression. And however, notice also that most of the species here are from African lakes. And so this gives you insight about how the light environment is actually the one driving this opsin expression. And this is because visual palettes are believed to maximize light detection in the light environment. So this is where there is a fish in a clear ecosystem. Notice how UV, violet, blue, and green wavelengths are transmitted very well. And also here in light intensity plot, you can see how shorter wavelengths are very abundant before 500 nanometers, before 450. And this is very different in a murky water environment because all the suspended particles in this water environment will absorb short wavelengths. And then here, notice how there is actually very little short wavelengths. And then if you plot the visual sensitivities of the fish, it's like, oh, they maximize light absorption according to the light environment they live in. So these also, we can also see this in data on MSP that we have from Cycla monoculus. So this horizontal range, these horizontal lines is the range of visual sensitivities of this fish. And then we have downwelling irradiance or light intensity from 3 Amazonian rivers. And this is the receding season, and this is in the rising season. And you can see how in both seasons you have short availability, little availability of short wavelengths. However, you also see that fish is trying to stretch a maximum. Usually fish or other animals don't have pigments absorbing more than 600 nanometers. So this fish clearly is stretching its maximum. And also notice how basically it's absorbing most of the light that is available in that ecosystem. And now we previously saw data on opsin expression. This is data on visual pigment sensitivity, which is data obtained through MSP. So here we have on the x-axis the wavelength spectrum, on the y-axis the different species of cichlids that have been analyzed throughout the years. And here you have the African lakes and neotropical rivers. And this data also is in agreement with our opsin expression findings because you can see that only species from Lake Malawi basically have UV pigments, sensitive to UV pigments, and also notice how neotropical rivers species actually are shifted toward red parts of the wavelength spectrum. So what we have learned about this is that Amazonian cichlids have lost and activated opsin genes, and this has occurred independently in different lineages. Amazonian cichlids have a long palette, which is based on the expression of the LWS2A, RH2A, or LWS, or basically blue, green, and red. And also that photoreceptors, spectral sensitivities are product of a long palette, and which are the most suitable for the light environment. So now that we have learned more about neotropical cichlids, I wanted to see if we could apply what we know about them, about the genetic basis especially, to other groups of fishes. And these are carassiforms, which are a very important group of fishes in the neotropics. And for this, I went to Panama to work with Michele Pirotti. He's a scientist at the Smithsonian Tropical Research Institute there. And this is how they look in the Panama streams. They are really, really fast and really hard to catch sometimes. And carassiforms are really interesting as well as cichlids. They also have a similar distribution. There are more than 2,000 species with one family that bulks most of this diversity with 1,700 species. They are also known in the aquarium trade as tetras, silver dollars, piranhas, neon tetras as well. And I wanted to understand how has evolution shaped the visual system of these fish. And for this, we went to Panama and we captured different fishes. In total, we captured multiple species, at least 13, including some species from Suriname as well. And they belong to 8 different families. And we did transcriptomes, 3 transcriptomes per species, and we extracted their opsins genes as well, and that's what we were interested for. So this is a fish opsin tree. This is based on the transcriptome of this species, but also from 2 species which genomes are available. And here you can see, so this, any collapsed color clade is different species from carassiforms, whereas all the others are like zebrafish and goldfish and lamprey, etc. So first, notice how carassiforms seem to have lost, all of them, this UV opsin, or SWS1. Also notice how they have duplications, and in LWS and in RH1, meaning the rhodopsin. And also how they still have representative sequences of the SWS2A and the RH2 opsin. So what it also seems to have happened is that these LWS duplicates are product of a teleus-specific genome duplication. So here we have our spotted gar, which is a fish that is known that didn't undergo that event. And we have here the Asian arowana, for example, and this fish and the rest of osteoglossiforms actually plot together with this second copy carassiforms, whereas we have all the other teleus and the LWS1 copy of carassiforms here in this other clade. And for example, if you look the opsin of the zebrafish, you see that it only has 1 sequence, whereas for some reason, carassiforms and osteoglossiforms retain these 2 copies, which is pretty interesting. And also what seems to have happened is that LWS2 and LWS1 underwent subsequent rounds of duplications only in the carassiforms. So here, for example, you have different clades for the LWS1-1 and 2-2, because this point is getting like kind of crazy. And here you have also, you cannot see it, but there are different duplications for each species, especially for each family, because remember that we're also analyzing through family. So this is very interesting because besides genes being duplicated after the genome duplication, subsequent rounds happen. And here you can see also that RH1 duplicates are also product of this teleus duplication. Here we have a spotted gar. However, arowanas did not retain a second copy. However, zebrafish did. And these zebrafish copies that are known to have been surviving copies of this event also cluster with the second copy of carassiforms. And here we have the other teleus. So this allow us to analyze the visual system of this fish from a opsin gene repertoire point of view. So here we have the species, here we have the family from which they belong, and here we can see their opsin. So here you have each opsin class. Here you have that you can see that they all lost the UV opsin. They all have their SWS2 opsin. Some of them seems to have lost or inactivated the RH2 opsin, the green opsin. And they have several copies depending on the lineage of the LWS opsin gene. So if you remember our great opsin protein and the 11-cis retinal like sensitive molecule chromophore, so what we found is that while analyzing this is that this second copy, LWS2, has 3 amino acid substitutions here that shift the maximal absorption of the opsin protein. So this changes at least 30 nanometers in absorption. This is pretty much from years of work from other visual scientists. And this is very interesting because these are the same substitutions that humans have in their LWS duplicates, which allow us to be sensitive to green and red. So that is pretty cool. So basically this new opsin gene acquired green sensitivity in this fish. So now this is carassiforms cone opsin expression. So first here you have the families and the species. Notice how all species express the SWS2 and how all of them highly express 1 copy at least of the LWS1 opsin and the LWS2 opsin. And notice how they are actually decreasing the expression, or they have little expression of the RH2 opsin. Which suggests that as one opsin acquired green sensitivity, the other opsin which had green sensitivity started to decrease in its expression. And this gene expression and sequence analysis are in agreement with carassine's photoreceptor sensitivity. So we did MSP on 7 species of carassiforms. And for example, you can see in this one, Bricon chagrensis, that its visual sensitivities match its gene expression. So we have this fish, for example, having visual pigments in the red, 3 parts of the yellow, green, and one in the blue part of the wavelength spectrum. And throughout our MSP sample, this makes sense based on gene expression. So that was also pretty great because it suggests that these fish are maybe more than trichromates and maybe even pentachromates. And this also makes sense in light of MSP, and this is previous data. So here we have autophytens. I referred to gymnotiforms and siluriforms, and here you have each of these is a different carassiform, different fish species. And you can see also the carassiforms. This data actually is in agreement also with our gene expression data because we do not have pigments absorbing the UV part of the wavelength spectrum, and also you can see that they are kind of like red shifted fish. And I was just here also I put siluriforms and gymnotiforms, which are catfishes and electric South American knife fishes, because it seems that the habitat is driving this as well, again, these visual sensitivities. So what we have learned from this is that the visual system of neutropical carassiforms, that we have different duplications, which are product of teleospecific gene of duplication event, and further lineage-specific duplications, and also that the LWS2 opsin, this pigment seems to produce a green-sensitive pigment and is highly expressed in the retina, which is also an example of gene neofunctionalization. And as because it seems that as one of the LWS genes became green-sensitive, it probably replaced the function of RH2. But that is still what we believe. So if we want to wrap up a little bit, here you have so the color vision as a trait. So the mechanism from an approximate point of view is that cyclists achieve color discrimination through color opponency, which is product of the interaction of 3 spectral channels. Also, that fish have spectrally different photoreceptors, which are product of the expression of at least 3 different opsin classes, and they require this to have color vision, at least these fish. They have several spectral tuning mechanisms, among which amino acid variation yields green sensitivity in carnosines. And opsin genes dynamics, so duplications, you have seen how these duplications in opsin genes, and also there are some opsin genes that get lost. These all modulate visual pigment diversity. So it's a mechanism in which color vision is also getting a lot of diversity. And then from an ultimate point of view, what is the adaptive significance of color vision? So fish exhibit behavioral color vision, which is essential in mating, foraging, etc. And fish visual systems adapt to the available light thanks to the different spectral tuning mechanisms they possess. And throughout the phylogeny in opsin genes dynamic, they highly vary throughout teleos lineage, as well as this opsin gene dynamic sometimes can lead the acquisition of new functions, like for example this one, the green sensitivity or differential expression, as sometimes opsin will start to be expressing in different ways after they have been duplicated. With that, I would like to thank everybody, especially my committee, especially all the members of the secret labs, past and present members, because they have they have helped me in taking care of my fish, and also all the other people that I have worked with and collaborated with, like has been great, and also the person that you all know has made all of this possible, the best advisor that I could have ever had. I could have never have asked for somebody better. Karen. And with that, I would also like to thank
So there's time for a few questions if anybody has some.
So that case of LWS2 gaining green sensitivity seems really interesting in light of it probably being convergence, like the case. So how many historical trajectories do you think are available to gain something like green sensitivity?
Yes, so we're actually trying to understand a little bit that. It seems that all of these, there are 5 sites in the amino acid sequence of these opsins that are the ones who majorly determine green sensitivity, and they seem to change in other animals as well. However, yeah, the fact that all of these 3 are there, at once is pretty interesting. So I was analyzing them through with the arowana data set too, with the osteoglossiforms, and it seems that after this replication, the previous ancestor already had this previous gene already had all of these 3 substitutions. Now, which one occurred first, I might not be able. I do not have a lot of idea of how it occurred. However, it seems that, for example, there was one that is more sensitive to. Maybe I can pull it up. This one. So we see that there is variability on this one, and this makes a little bit of sense because it's 7 nanometers and not such a big change as 16 nanometers. So for example, there is more variability in this one, so I would imagine that this would be a very variable change. So maybe this one occurred first, for example, because the likelihood of happening may be higher.
And do you think that this this gain will always be preceded by loss? So you have this interesting thing where RH2 is then lost, right? So is this predictive? Can certain neofunctionalization events possibly be predictive? To my knowledge, no one's shown that. But
yeah, so it's all about how do you decide to get rid of this, right? Because for some reason, some fish keep it and other fish just get rid of it. So I guess there is first selection on low expression, and then for some reason it gets lost because there is no selection to preserve that specific sequence. But I guess it all starts with less selection for expression of one of the opsins. Yeah, but it's interesting because these fish also express the RH2 still. So that means so I don't know how maybe some gene regulatory mechanism is making LWS, the screen sensitive LWS, be more expressed than the RH2 opsin. Yeah, and also these, I didn't mention this, but we believe that these ones have a sensitivity around 500 nanometers, and not just 530. And however, the RH2 green opsin has a sensitivity around 490. So maybe for the fish it is still important to keep this. Yes, Julie.
So back to your second chapter, looking at the Amazon and African cichlids. Obviously, the habitat is super related to their spectrum, visual sensitivity, and obviously that's been selected for. What aspect of natural selection do you think might have been more responsible for that within the African cichlids? Is that mating or sexual selection? Or,
the fact that
or
African cichlids have retained.
predation? Well, just obviously it shifted from red to more, the shorter wavelength. So what would have been that environmental pressure?
I would say that foraging in clear environments. So it has been associated UV sensitivity for a lot of fish that eat plankton and zooplankton, because according to that theory, plankton with UV light, there is a lot of contrast between the space light and the plankton. So the fish that have a short palate are usually fish that eat that eat plankton and other, I think phytoplankton too. So I will say that, and also because in a clear environment, many of these fish of the short palate species live kind of in shallow waters with these shorter wavelengths are pretty abundant, whereas cichlids in Lake Malawi, in deeper waters, they tend to have a medium or long palate because those short palettes those short wavelengths are already gone, and only green and blue is kind of transmitted best. Yes.
So I'm really struck that the New World fish, the kerosines, they have 5. This pentachromatic idea. So if you think about that just kind of as a modeling, and you've done some of this, are they expected to have a lot more sort of sensitivity, discriminating closely spaced wavelengths, and do you think that's what's driving it, or is it?
I don't think so.
Yeah.
Many fish may have different visual pigments, but that doesn't mean that they will be better at color discrimination. Also because they have these visual sensitivities, but we don't know how they are being compared in the brain, or how they are being averaged. And also they may have we do not know anything about if this is in a region in the retina, or this is in the central part of the retina. Many times fish have kind of a phobia, part of where the photoreceptors are very highly concentrated, and usually there they only have 3 different visual pigments, and that's it, because that is the optimum for color discrimination. However, because for MSP, you basically take out the retina and you shred it into pieces, and then you put it there, and then you like just, if you are lucky to find a photoreceptor, then you measure it. So you don't know anything about where it was or like how it was. But yeah, I mean, it's cool that they have different types of photoreceptors. Like that is really, really amazing. However, we do not know how they are using this
More to be done.
to see color. Yeah.
So, for your behavioral experiments, it looked like from the names you had males and females. Was there any difference?
Yeah, so the first ones
Sexes?
were males, and the other ones were all males, and the other ones we still do not know. Yeah, but regarding opsin expression studies, they do not have differences in opsin expression, so we wouldn't expect differences in their performance. However, males are more, since males are more territorial, sometimes they are better at like training and stuff because they are less afraid of you. However, both females and males, they will need, like males would need great color vision not just for foraging, but also like for identifying other dominant males. Whereas females will, according to the sexual selection, they will also need color vision to discriminate between males from their species and dominant and subordinate males.
If there are no further questions, we'll thank Danny for a great